Hello. Hi, Karim.
Hey,
Hi.
good morning, Donia.
How
Danya.
are you doing well?
Yeah.
Hanging in there. How about you?
A lot of work. Me too, hanging in there.
I know, right? That's the name of the game, just hanging in there.
I mean, we had a
What I
lot of.
know, what the one thing I want to find out, and is Isabella on right now? She just has her video off. Um, are we as so we want to make sure, Sheree, that I think that you and I as panelists that our videos don't show up while she's talking at any rate. And I don't 100% know the settings. Like if everyone's a panelist, do we automatically as a panelist show up? Do you know?
I
Once we start
think
the
if we
webinar?
do, let me try one thing. Let's see. So if we turn off our videos, then I have
Then
Donia
we just
centered
go
now.
away. Say that again.
So if I do this, if I say spotlight, Donia,
Okay.
then I think we can just, let's try, we can shut off our videos and then, I mean, we still show up along the top.
Yeah.
I think it's
Okay, you think it works? Okay,
No,
and then
but I
I don't.
think also if you have your microphones muted,
Yeah.
then you shouldn't pop up in the because I will be sharing my screen, right?
yeah,
So
exactly. So I think it'll be you.
Okay. Where did you
so
even see the spotlight function? I don't even see that.
Let me see if I take it off, if you can see it. If you go to like the participants, open that little panel on
Yeah.
the side, click
Yeah.
video, like hover over the video where it says more,
Yeah.
and then drop down, it says spotlight for everyone.
Oh, no, mine just says chat or pin,
Huh. Oh, okay.
actually. So,
Maybe that's because I'm a maybe you're not a co-host or
oh,
something.
no, maybe not. Maybe she just made you because you were, yeah, hosting originally. That may be. Anyway, I was just curious. Now I understand why I can't see it, which is totally fine. Um, but
Yeah.
obviously, Danya, you can see we've hosted so many webinars before that we're
yeah.
really proficient at this. So unfortunately, you're our guinea pig of doing it on our system for the first time, but I'm sure it will all be fine. There are like 70 people registered, so there was
Wow.
a lot of interest.
Yeah, I know that some of my colleagues from here at Mount Sinai, they will be joining too.
Okay.
Great.
So I think you have to make me co-host so I can share my slides. And let's try that to make sure that it shows up
Yeah,
well.
that's a good idea.
Okay.
Okay, so it seems now I can share. Let's see if that works. I need to put my presenter view. Can you see my slide well?
Yeah, that's perfect.
Okay, that's great.
Isabella's coming on. We can ask her a question, see if she knows. Isabella, when Danya starts talking, for those of us, like the rest of us who are sort of shown as panelists on the side, is there a way to make us disappear from view, or do we just need to go off camera and just pretend not to be there?
That's a really good question.
Do you happen to know? I mean, I guess I don't want to screw things up because sure, at the end we need, if you're going to do this, we need to facilitate question and answer. So at the end of the day, if we're just off camera and Danya's spotlighted, that's probably sufficient.
Yeah, I think it'll probably be okay.
And then also at some point,
yeah, see if I spotlight her, then she's the only one that shows up in this mode.
okay, and we have attendees joining. Can they actually hear us figuring all this stuff out? Probably.
It's okay, I see all friends, right?
Let's see. Hi, everyone. We're just waiting for another moment for people to join. Looks like we have a good number of participants this morning, early afternoon, wherever you may be. So yeah, it looks like time. So maybe we'll get started. Great. Let's see. So thank you everyone for joining us today for our April ISEE North American Chapter Seminar. Please join me in welcoming Dr. DANIA VALVI. Dr. VALVI is an assistant professor in the Department of Environmental Medicine and Public Health, the Icahn School of Medicine at Mount Sinai in New York. Prior to joining Mount Sinai, she worked as a research scientist at IS Global in Barcelona and completed her postdoctoral fellowship in environmental health at the Harvard T.H. Chan School of Public Health. She holds an MD degree from the University of Crete and an MPH and a PhD in biomedicine and epidemiology from the Pompeu Fabra University in Spain. Her research focuses on the impacts of developmental and life course environmental exposures on human health, especially on initiation and progression of metabolic diseases such as obesity, diabetes, and chronic liver diseases. And her recent work has focused on the application of omics approaches to facilitate the understanding of disease mechanisms. And today she'll speak about her work related to PFAS exposure and type 2 diabetes risk across the life course, links via the human metabolome. So with that, please join me in welcoming DANIA VALVI. Thank you.
Thank you, Sarah. Thank you for the invitation and also for the nice introduction. Can you hear me well?
Yes.
Okay, great. So, and thanks everyone for joining us today. It's a great pleasure to have the opportunity to talk about PFAS and diabetes and share with you results from our recent studies integrating metabolomics with the aim to better understand mechanisms of PFAS metabolic toxicity in the population. So this past week was actually the Diabetes Alert Day in the United States, and you may have noted a lot of talk in the social media about the diabetes pandemic. And for good reasons, because this is one of the most serious public health epidemics that we face globally. Here we see key findings from the International Diabetes Federation global report that was released this past year. There are more than 500 million adults globally who are currently living with diabetes. This is equivalent to 1 in every 10 adults. And if we do not come up with better prevention strategies, these numbers will keep rising, exceeding the 700 million diabetes cases in the next 10-20 years. Another 500 million of people globally have what we call impaired glucose tolerance and pre-diabetes, which puts them at a significantly higher risk for developing diabetes in the next few years. And this is an important public health crisis because diabetes not only impacts the life quality of patients, it increases the risk for many other severe chronic diseases such as heart disease, kidney disease, nerve damage, hearing and vision loss, as well as mental illness, but it is also a leading cause of death, especially in middle and high-income countries. And we may witness even faster increases in the prevalence and incidence of diabetes in the next few years because of the COVID pandemic. So very early in the COVID pandemic, it became very clear that people with diabetes and hyperglycemia had much worse COVID outcomes compared to healthy individuals. But we now understand that this relationship is much more complex and also bidirectional. So now we have evidence that coronavirus can actually directly impair beta cell function and may increase the risk of new onset diabetes after COVID. We also start to see that the COVID pandemic have may have multiple indirect effects on the diabetes epidemic, and especially through the impacts of lockdown and social distancing measures, for which we have the first studies on longer-term effects to show that these measures have likely contributed to further increasing the global prevalence of diabetes. North America, and especially United States, is one of the global regions with the highest prevalence of diabetes, which is not surprising considering also the epidemic rates of severe obesity that we see here. Obesity is a very important risk factor for diabetes, but it's not the only factor. And we have been seeing increasing rates of diabetes even for population subgroups that are within the normal weight range. In the US now, there are more than 37 million people with diabetes. Over 90% of these cases are actually type 2 diabetes, which is the most frequent type. But we have seen also similar increases in pediatric diabetes type 1, as well as gestational diabetes. And even though US has one of the lowest prevalence of undiagnosed cases, 1 in every 5 diabetes cases remain undiagnosed, which is a very important barrier for diabetes management and also for the prevention of diabetes complications in the population. Another very important issue are the very big racial and ethnic disparities that we see in diabetes. Diabetes is a disease that disproportionately affects ethnic minorities, especially Native Americans, Blacks, and Hispanics. These ethnic minorities are more likely to develop diabetes, but also to suffer from complications and to die because of diabetes. We know that these disparities are in big part due to inequalities in socioeconomic factors and the accessibility to healthcare, but we don't fully understand these disparities. And in ISEE, we know that there are also important issues of environmental justice that are less appreciated. And there are, for example, differences in environmental chemical exposures and exposure mixtures across racial groups that could also be a factor contributing to these health disparities. So PFAS is a class of per- and polyfluoroalkyl substances that we know that can interfere with the hormone system. So they belong to the broader category of endocrine disrupting chemicals. And PFAS have attracted lots of scientific interest in recent years for many good reasons. One of them is because this is a particularly big class of chemicals now released in our environment. There are over 12,000 compounds that are estimated to fall into the PFAS category, and this number is continuously rising. PFAS are also very mobile in the environment, and this is why they have been detected in almost all parts of the world, even in the Arctic, far from industrial sites. So we are currently facing a widespread global contamination PFAS problem. In addition, we know that many PFAS are very highly resistant to degradation because they have many carbon-fluorine bonds. As you can see here, I'm showing PFOS and PFOA. These are two of the most widely studied PFAS. Carbon-fluorine bonds are very strong and stable. They do not break easily down. And this is why sometimes we refer to PFAS as forever chemicals because of their extreme persistence in environment. So once PFAS are released in our environment, they stay there for years and they accumulate in the food chain and humans. As a result, human biomonitoring studies show that more than 99% of people tested have some mixture of PFAS in their blood. This has been shown in NHANES and also populations all over the world. PFAS have been widely used in several consumer products and other applications since the 50s. However, it was not until the early 20s that we started figuring out their impacts in our health. And that was in large part because we started noting that there is high detection of PFAS in drinking water, especially here in the United States. There are multiple ways that PFAS are released and transported in the environment. A major one is from the direct release, of course, of PFAS in groundwater, the atmosphere, and solid waste from industries that produce or use PFAS in their manufacture. And because PFAS are very mobile in the environment, from there they can transfer to other parts of the world. So the most effective way to deal with this widespread exposure problem is to limit the production and use of PFAS in the first place. And there have been increasing efforts from policymakers and stakeholders in recent years. You may have heard, for example, that EPA announced 2 weeks ago its proposal for the first ever national standards to limit PFAS in drinking water. The European Union is further ahead and is also moving towards regulating more than 10,000 PFAS as a class. But it will take at least a few more years until we see the strict regulations that we need to effectively reduce PFAS exposures. And because of their environmental persistence, I think PFAS will likely continue being a public health threat for many more decades, similarly to what we have seen in the past for other persistent organic pollutants, even though that have been previously regulated. So the most common routes through which we are exposed to PFAS is by consuming contaminated drinking water, contaminated food, and through the use of everyday products that contain PFAS. Studies show that dust inhalation and dermal absorption can also contribute to a lesser extent to the body burden of PFAS. So PFAS, because they have water, oil, and stain-resistant properties, they have been broadly used in several everyday products, such as non-stick pans, fast food packaging like pizza boxes, waterproof clothes, water and stain-resistant carpets and furniture that we may have in our home, floor treatment waxes, in dental floss, certain cosmetics. Recent studies found PFAS even in period underwear, and the list goes on and on. And because PFAS are found in so many everyday products, this makes exposure very hard to avoid. And this is why we need strict regulations to be able to effectively reduce exposure in the population. Some individuals also have high exposure to PFAS through their occupation. For example, fluorochemical workers and firefighters are two occupational groups in which we have seen high exposure levels in studies so far. PFAS exposures have been associated with many adverse health effects, not just metabolic effects. We have good evidence from both experimental and epidemiology studies that PFAS, for example, are associated with intrauterine growth restriction and low birth weight, immunotoxic effects, and reduced response to vaccination in children and adults. And we know that PFAS act also as thyroid disruptors. Data on cancer have been more limited. However, studies in highly contaminated communities, such as the C8 Science Panel studies in Mid-Ohio Valley cohort, show associations with testicular and kidney cancer, among few other cancer types, which is also further supported by evidence from in vivo models. PFAS can also alter the reproductive system. We just published a study with our collaborators from Singapore using data from the preconception espresso cohort, where we found that PFAS is associated with reduced odds of pregnancy in women who were naturally trying to conceive. Previous studies have further shown similar decreases in fertility in men and also in couples, which I think are very concerning results. And I think this is a very important field for future research related to PFAS. In regard to metabolic effects, we have good evidence that PFAS increases cholesterol and blood pressure in humans. We have some mixed results for diabetes, for which I will talk more in a bit. And in a recent systematic review and meta-analysis of both rodent and human studies that was led by my collaborators at the University of Southern California, we found consistent evidence that PFAS exposure increases ALT, a liver enzyme, suggesting that PFAS is a potential risk factor for non-alcoholic fatty liver disease, a disease for which we have also seen epidemic trends in recent decades. This is a meta-analysis on PFAS and type 2 diabetes that was published a few months ago. On the left, we see the results for the association of PFOA. On the right, the results for PFOS. After stratifying studies by study design. And there is a lot of heterogeneity in effect estimates across cross-sectional studies and case control studies with some inconsistent results. But when we look at prospective studies, it becomes more clear that exposure to PFOS and PFOA is associated with increased risk for type 2 diabetes. Prospective studies conducted so far in this field have been mainly in high-risk population subgroups. For example, the two studies by Andres Cardenas and collaborators were in the Diabetes Prevention Program trial, and all of them had overweight and obesity. Kearns and collaborators studied PFAS association with type 2 diabetes in the highly exposed Mid-Ohio Valley cohort, and the study by Park and collaborators is in SWAN, an historical community-based prospective cohort study of midlife women. And I think it's the largest study that we have to date, with about 1,200 women who had no diabetes and have been followed to later ages. These are all very important studies that support the hypothesis that PFAS are diabetogenic in the population. And now there are a couple more prospective studies in this field. But looking at the available literature, I think, and thinking about what we can do to strengthen the evidence further, I think a few things that we can do are to confirm also results generalizability across, more broadly across population strata, is to conduct more prospective population-based studies that are ideally larger and diverse in terms of race and other socioeconomic factors, which can allow us also to better understand some of the disparities that we see in diabetes. We also need more life course approaches that can give us a hint about potential windows of enhanced vulnerability. So far, we have been focusing in evaluating also a limited number of PFAS. We definitely need more work, including also recent PFAS replacements. And we are also lacking data on how PFAS may interact with other known risk factors for type 2 diabetes, for example, with our genes or certain lifestyle factors, which can be important for informing the design of interventions. We also now have unique opportunities for understanding biological mechanisms through the integration of omics in our epidemiology study designs. One example is metabolomics, for which I will show you results today. But of course, there are many other interesting omics layers. And as we move forward, I think an interesting goal for our field is to extend our methods and move towards more multi-omic approaches, which may allow us to get a more holistic characterization of mechanisms of PFAS toxicity at a population level. So now I'm going to show you results mainly from two prospective studies that I have been working with my collaborators with the goal to address some of these gaps. I'll first show you results from the earlier studies we did in the oldest Faroese birth cohort. This is a population-based prospective cohort that was initiated back in the 80s, and now it has been followed by my collaborators, Philippe Granzan and Paul Wave and colleagues towards adulthood, where we had the opportunity to study multiple early life exposure windows. And then I'm going to show you some first results from the nested case control study of incident type 2 diabetes that we are now doing within the multi-ethnic BioMe biobank. This is a very large biobank with more than 65,000 patients enrolled here at Mount Sinai since 2007, who are fully genotyped. We have rich phenotype and geocoding data from their electronic health records, as well as stored plasma samples from enrollment that we now use to generate exposomics data. Each one of these studies has their unique strengths and, of course, some limitations, like all observational studies, but taking results together, I think they can offer good complementary insights about the potential impacts of PFAS on the development of type 2 diabetes over the lifespan. So we'll start with the Faroese cohort study. Here you can see the study design. Participants have been followed every 7 years since their birth towards age 28 years. The Faroese are a fairly homogeneous community in terms of socioeconomic and lifestyle factors. It's a small fishing community with high seafood intakes. One of the traditional foods has been a pilot whale, from where much of their exposure to environmental contaminants comes from. So exposure to 5 major PFAS, mainly long-chain legacy PFAS, were measured in this cohort in whole blood and also in participant serum at later ages. And metabolic assessments were performed at all examinations with the inclusion of a 2-hour oral glucose tolerance test that was performed at the latest visit at age 28, from where we calculated indices of insulin sensitivity and pancreatic beta cell function to investigate the PFAS association with the risk of type 2 diabetes. For all PFAS, we found higher concentrations on average in postnatal examinations, and especially at ages 7 and 14 years, which is in part attributed to the PFAS accumulation through breastfeeding, breast milk, in combination with the long half-life in biological tissues that these long-chain PFAS have. On the right, we see the individual trajectories for the concentrations of PFOS, where we note a decline in exposure after the 14-year examination and through age 28, which is in line with the decline in environmental PFOS levels we have seen after the early 2000s, when the industry started to substitute PFOS by other PFAS replacements. So we examined the association between individual PFAS across all ages with the indices we calculated from the OGTT. And what we mainly found was that exposure to PFAS across all ages is associated with a decrease in insulin sensitivity. These are the estimates that we see on the left. And effect estimates were fairly comparable, as you can see, across examination windows, suggesting that both prenatal and postnatal exposures may be contributing to a decrease in insulin sensitivity over the life course. We also found that PFAS exposure, and especially exposure occurring prenatally, is associated with an increase in insulin secretion from the pancreatic beta cells. These are the associations we see here on the right between PFOS and the insulinogenic index. We did not find significant interactions by sex. However, for some associations, we observed stronger associations in females compared to males. But our results overall from this study suggested that both sex can be affected by PFAS exposure. Our participants were still very young. By age 28, less than 8% had impaired glucose tolerance, and almost none had type 2 diabetes. So we were not able to confirm a more direct association between PFAS and diabetes. For this, we will have to wait for the cohort to reach later age. But what our results suggest is that PFAS exposure may decrease insulin sensitivity and at the same time can contribute to a compensatory increase in insulin secretion by beta cells, which from the pathophysiology of type 2 diabetes, we know that it's a mechanism that on the long term, at least in some individuals, it can lead to beta cell damage and diabetes. We also used the structural equation modeling approach to estimate lantern functions of life course exposure to each PFAS based on measured concentrations across the multiple examination periods, and we associated this to a latent function, an estimate of glucose homeostasis based on multiple type 2 diabetes indices that we calculated from the OGTT. And results from the same analysis further showed that in our population is mainly PFAS exposure that is associated with altered glucose homeostasis at age 28. Estimates for PFOA and PFH excess were in the same direction as for PFOS, but not statistically significant. The exposure range also for these other PFAS was more narrow, which may be one of the reasons for which we found associations mainly for PFOS. However, in other studies that we have conducted and studies that conducted by others, we have seen associations also for PFOA and PFH excess among other PFAS. In a follow-up study, we examined whether genetic predisposition to diabetes may modify the association of PFAS with insulin sensitivity. So we used a targeted SNP approach, where we studied a total of 85 SNPs that had been selected based on previous reported associations with type 2 diabetes, out of which 5 SNPs passed our false discovery rate correction threshold for a potential interaction with either PFOS or PFOA in association with insulin sensitivity. Here, for the sake of time, I'm showing only the results for PFOS, which include variants in the ABCA1 gene, that is a key regulator of lipid transport and metabolism. 2 variants in the FTO gene, that is strongly associated with obesity and regulates adipogenesis and energy homeostasis, and a variant in the PPR gamma gene that is implicated in adipocyte differentiation and glucose homeostasis. Of course, we need to replicate these findings in other independent populations. That was one limitation of our study. But these findings were of great interest to us because they suggest potentially synergistic effects between PFAS and genetic predisposition to type 2 diabetes. And if are replicated in other settings, these results can be informative for the design of personalized interventions. So in recent years, we have also been working with my collaborators in the integration of untargeted metabolomics with the goal to understand better mechanisms of PFAS toxicity in the population. We recently conducted also a systematic review on this topic using PRISMA guidelines. So we reviewed epidemiology studies who have evaluated the association of PFAS with either targeted or untargeted metabolomics. Sandra, a postdoctoral fellow in my group, did the fantastic work in putting all this evidence together. So we identified 28 eligible studies. Almost all of them were cross-sectional. There were only 4 prospective studies included in our review. Most studies were in adults, and almost all of the studies had measured metabolomics at a single point in time. And we did find some consistent findings across studies. Here in this figure, I'm showing a summary of pathways based on the KEGG database that have been associated with PFAS in 3 or more studies. There are more than 100 pathways reported in previous literature to be associated with at least one PFAS. And most of them, as we can see, are involved in amino acid metabolism, for example, glutamate and arginine and proline metabolism, or lipid metabolism, and especially glycerophospholipid metabolism. And there are also quite few pathways implicated in carbohydrate metabolism and the metabolism of vitamins and cofactors. Many of these pathways have been further shown to be associated with type 2 diabetes in large metabolome-wide association studies. We also performed our own independent pathway enrichment analysis using Reactome to more systematically synthesize prior evidence in this field. Reactome is an open source peer-reviewed pathway database for metabolomics data. So we extracted metabolites previously associated with PFAS to identify in which pathways these metabolites mostly were involved. And here we see the pathways that passed the FDR threshold. And this included several transport of small molecule pathways, such as amino acid transport and lipoprotein remodeling pathways, as well as pathways of transmembrane transport and associated disorders, such as the SLC transporter, the solute carrier transporter, pathways that are strongly associated with diabetes, but also with other metabolic diseases. And these pathways are now supported by a large number of previous studies. Of course, results get less consistent across studies when we look at its specific metabolite or the directionality of associations. But I think the evidence that we now have in this field is very exciting, as we have just started to understand mechanisms of PFAS toxicity through the study of the metabolome. We also noted some important limitations that I think we should think more in future studies. The one is the potential of reverse causation, because almost all of this evidence comes from cross-sectional study designs. There are also concerns about confounding by concurrent exposures that may affect the metabolome, such as diet, for which we have not controlled very well so far in our studies. And we are also lacking longitudinal studies with repeated metabolomics measures, which can help to understand to what extent associations reflect also long-term effects and perhaps persistent effects on the metabolome over the lifespan. So to address some of these limitations, we went back to the Far East cohort and used serum samples collected at the age of 7, 14, 22, and 28 to generate repeated measures of untargeted metabolomics. We performed this analysis with Dean Jones and Ryan Smith at Emory University, and we were able to get complete data and measure metabolomics across all ages for a total of 125 Far East participants who had sufficient serum volumes from the childhood examinations. This work was funded by an NIHS R21 exploratory research grant, where one of our additional goals was to leverage previous causal inference methods to strengthen the evidence for potential PFAS effects on the human metabolome and its implication for human disease. So we have used the causal inference framework developed by Donald Rubin and extended by my colleague and good friend Mariabel Bint. In this case, this framework has already been applied by Mariabel and other investigators in environmental epidemiology studies in the past. Here you can see some of the papers. And it has been applied using other omics layers, for example, microbiome data. So here the novelty is that we use this framework to investigate potential PFAS effects on the metabolome. Causal inference approaches have been scarcely used in the PFAS field up to now, and I think can be helpful in this context, especially because it is very hard in the case of PFAS to have actual randomized controlled trials. RCTs, PFAS are also fairly persistent, so it's very hard to design an actual intervention. So what we can do instead is to use already developed causal inference methods that can help us emulate an RCT based on the observational data that we may have. And of course, our study in the Faroes, it was a small size study, it's not going to answer all the questions, but I think it can serve as an example of how we can use these approaches more broadly in this field in the future. So we were interested to answer the question whether high versus low PFAS exposure altered the metabolome. And we were especially interested in prospective associations, so on the long-term and potentially persistent effects that early life PFAS exposures may have on the metabolome. So to answer our question, we used our observational data to reconstruct hypothetical randomized experiments as if we had applied a randomized PFAS increase intervention to our participants. So we started by classifying our individuals into two groups of PFAS exposure, high and low, using median exposure cutoffs. And we made sure that our two groups have similar balanced covariate distributions. So we used pair matching for that to handle individual specific confounding, considering a wide list of confounders that we selected based on a priori knowledge using directed acyclic graphs. We did that separately for each PFAS at each window of exposure. Of course, we are also interested in mixture effects. But here, considering the sample size restriction, we decided to keep things simple for our own analysis and designed our experiments based on individual PFAS exposures. So once we designed our experiments, we moved on to analyze the data and test the null hypothesis of no effect of the intervention at different aggregation levels of our data to get our results. Vishal and Mason, that you can see here, did the fantastic work and all the heavy lifting with the data. We are now finalizing this analysis. So the results that I'm going to show you today are still preliminary. One challenge that we had with this analysis is that it is very computationally very intensive, and we are still working on running some sensitivity analysis. So these are some preliminary results. So here we see plotted the randomization-based beta estimates only for those metabolites who survived multiple comparison correction in at least one examination age, in association to either prenatal PFAS exposure, this is what we see in the first plot, or postnatal PFAS exposure at the age of 7 years. This is what we see in the plot in the bottom. And if a metabolite survived multiple comparison correction in at least one age, then we have plotted effect estimates for this metabolite across all ages to see whether this association may have persisted over time or maybe not. If an association has persisted, I think it can give us even greater confidence that this could indeed be a true and important long-term effect. So for prenatal exposure, results suggest associations mainly for PFOS. For example, we see an association with pantothenic acid, which is pretty consistent from childhood to adulthood, and also potentially an association with glutamate that it's more prominent at the age of 22. For postnatal exposures, we found evidence for associations with other PFAS, mainly PFOA and PFH excess. And in this case, we also see some fairly consistent results across ages. For example, PFOA seems to increase dimethylarginine post-puberty, and this is supported by both adulthood examination. We see this association at both adulthood examination periods. We ran also a pathway enrichment analysis using Reactome, including all PFAS-associated metabolites that popped up in our analysis. Here we included also those that were associated with PFAS exposure at age 14 and 22. I'm not showing here these associations for simplicity and because our results are preliminary. But what we see is that the metabolites that we identified are involved in some pathways that were also previously indicated in our systematic review. So our longitudinal study here in the furrows seem to support that at least some PFAS-associated metabolome alterations previously reported in adult studies may have started early in life and could potentially persist over the life course. And we were able to show that using a causal inference framework that give us a bit more confidence for potentially causal associations. Of course, we can never eliminate all sources of error or bias from observational data. And our study was also small in size, but I think if we leverage more broadly causal inference methods in this field, it can help us address some of the current limitations we have, and it can help us make stronger inferences about potential PFAS effects on the metabolome in the future. So now I'm going to show you some of the most recent work we have been doing on PFAS and type 2 diabetes with my colleagues at Mount Sinai, the University of Southern California, and Emory University, using data from the multi-ethnic BioMe Biobank. So since I started at Mount Sinai, I have been thinking about how can we address some of the limitations that we have in our previous studies and strengthen the evidence about the PFAS link with diabetes. And I'm very interested in diving deeper into ethnic disparities and what's the role of the exposome in these disparities. And this inspired an initial P30 pilot study that we conducted to generate preliminary data and investigate these aims in BioMe. Now we have gotten also R01 funding from NIHS, and we are expanding this work to a larger scale study. We are now in year 1, so we still have a lot of work to do, but I'm very excited to talk about our overall plan and show you some results that we already have from the pilot study. And here you can see all my colleagues involved in this project with who I have been very fortunate to work with. So BioMe is a very large biobank that has enrolled patients seeking mainly primary care at Mount Sinai since 2007. Some major strengths of BioMe is the wide ethnic diversity with a very good representation of minority groups that are disproportionately affected by diabetes, especially African Americans and Hispanics. Almost all BioMe participants have in-depth genome phenotyping. The initial actual goal for BioMe was to perform GWAS analysis in association to a variety of diseases. Diabetes has been one of them. And we also have very rich data through the electronic health records, as well as questionnaires that the participants completed at enrollment when they also provided a blood sample. So in our R01 investigation, we apply a nested case control design within BioMe, where we have selected 850 incident type 2 diabetes cases and another 850 matched controls. We are matching them based on age, sex, race, and calendar year at blood draw. We will measure PFAS and untargeted metabolomics in pre-diagnostic plasma with a goal to examine the association between the PFAS mixtures with type 2 diabetes, potential gene-environment interactions, and implicated mechanisms using high-resolution metabolomics. An additional strength of our study design, I think, is that we aim to replicate our results in the multi-ethnic cohort in Los Angeles and Hawaii, which will provide stronger support for our findings. Both cohorts are very diverse in terms of ethnicity, so I think this gives us a great opportunity to also dive into interactions by ethnicity to better understand the health disparities we see in diabetes. So here our focus is on both targeted PFAS, but also we will do an untargeted PFAS screening to study the perfluorome. For this, we are leveraging the state-of-the-art methods that Doug Walker has been developing in the past few years for capturing the perfluorome using high-resolution mass spectrometry. So we will be studying many more PFAS, including more recent replacements than what we have been studying so far. So for our pilot study, we measured PFAS and untargeted metabolomics in 180 cases and 100 matched controls. And what our results suggested is that there may be a 27% increase in the odds for type 2 diabetes per quartile increase in the PFAS mixture. Here, for analyzing the mixture effect, we used weighted quantile sum regression, which further estimates the contribution of its PFAS in the mixture association. And as you can see, our results suggest that major contributors to this mixture association are mainly sulfonic acids, PFOS, and PFH excess. In general, we found comparable PFAS exposure levels in BioMe compared to ENHANCE. So for most PFAS, so exposure levels are pretty representative of the general population. We did not observe a clear trend of interaction by ethnicity, but of course our pilot sample was underpowered. We will be able to examine this more in depth in the larger study. We conducted also a meet-in-the-middle approach to see what metabolites and pathways are associated with both PFAS and type 2 diabetes in our pilot sample. And as you can see, some common pathways that we found, for which we found associations with both the PFAS mixture and the odds for type 2 diabetes included two pathways of amino acid metabolism, glutamate and arginine and proline metabolism, and the pathway of drug metabolism, cytochrome P450. So we are now excited to test these aims in the larger BioMe study and in the multi-ethnic cohort in LA and Hawaii, where we will further examine gene-environment interactions and some more additional interesting aims. So in summary, some of the main conclusions that I think we can draw from our studies and also from the state of evidence from epidemiology studies up to date is first that PFAS exposure over the life course may decrease insulin sensitivity and alter pancreatic beta cell function. And in this way, it can progressively lead to type 2 diabetes in humans. We also note from recent meta-analysis that cross-sectional studies have generated very conflicting results, and I think this is something that we should take into consideration as we move forward. So I think what we mainly need now in this field is to have more prospective studies, ideally in larger and more diverse populations, to be able to strengthen the evidence for a PFAS link with diabetes. A growing number of epidemiology studies integrating high-resolution metabolomics methods have already started to uncover metabolic dysregulations linked with PFAS exposures. In our recent systematic review, we already identified some consistent results across studies, especially for pathways implicated in amino acid and lipid metabolism. But there is also a lot of noise and likely false positive, but also false negative results. And there are some limitations that we noted, for example, the cross-sectional design, which is prone to reverse causation bias, the lack of longitudinal metabolomics measures, which can help us to better understand long-term and persistent effects over the lifespan. Of course, there are concerns related to unmeasured and residual confounding, and something that I didn't talk earlier today, but there might be also some tissue-specific effects. However, the evidence that we have so far is mostly from studies that have measured metabolomics in peripheral blood, in some occasions urine. So tissue-specific effects is also, I think, an interesting field for future research related to PFAS and metabolomics. We can think also about leveraging more broadly in the PFAS field and the environmental omics field, causal inference methods. Such approaches have already more widely used in other environmental epidemiology areas, for example, in the field of air pollution. Of course, for this, we need also larger samples, but now we continuously do great team science efforts to harmonize and generate larger environmental data sets. For example, there is a lot of work on that on ECO and other large exposon projects that we have here in North America, but also in Europe, that can offer this opportunity for the future. And as a last point, I also think something very important is to do more research on potential effect modifiers. For the moment, we have very limited knowledge about potential factors that interact with PFAS in the development on diabetes and generally human disease. There are some great emerging hypotheses. For example, we found some interesting gene PFAS interactions in our Far East study. There have been also hypotheses about interactions by lifestyle factors. For example, physical activity has been proposed to mitigate to some extent the PFAS effects on metabolic disease. There is a growing interest also about potential PFAS interactions with diet. And these are all very important research questions that if we address, they can generate results that inform personalized interventions for the more effective prevention, but also management of diabetes in high-risk population subgroups. And with this, I would like to thank all these fabulous people from my group that have been involved at different degrees in the studies that I showed today. We are a very vibrant group. Working together is always a lot of fun. We also have now an open position for a postdoctoral fellow. So if any of you is interested in a postdoctoral opportunity and you are interested in this field of research, please get in touch. And why not come to join us at Mount Sinai? And of course, I'm very thankful to all my collaborators and colleagues from the Far East birth cohorts the BioMe and MEC multi-ethnic cohorts. This is a great team science effort, and it would be possible without all their valuable contributions. And of course, I would like to thank NIEHS for supporting our studies. And I will finish here, and I will be very happy to answer any questions you have.
Thank you so much, Dania. It's a wonderful talk. Sorry, I'm trying to figure out. Oops. Sorry, there we go. Webinar technologies. So please feel free to raise your hand or put your questions in the Q&A. It looks like we have a couple of questions in the Q&A to start. Let's see. I don't know, David, if you're on, please, I'm happy to unmute you if you'd like to ask your question. Let's see.
Yeah, sure. Okay. Question. It was interesting to me in your earlier slide, showed the prevalence of type 2 diabetes. I think it's now 11%, if I recall, and the dramatic rise in the US over the last 20 years. It was also interesting to me to note that over the approximate same time period, blood PFAS levels declined by more than 85%, and blood PFOA levels declined by more than 70%. So there have been large increases in type 2 diabetes over the last 20 years, but over the last 20 years, dramatic decreases in PFOA and PFAS in blood over that same time period. Any thoughts back on that? From
that's
you?
That's an excellent question. You know, I have been working on this endocrine disrupting chemicals field for a while now. PFAS, I have you know, focusing mostly in the last few years, but this is an interesting question that we have been receiving over the years for many of our studies, also for PCBs and TD, you know, more legacy persistent organic pollutants. So how do we explain the fact that these exposures over time may have been decreasing while the disease prevalences are increasing? And of course, you know, we cannot correlate these trends because we can, you know, if you see them at an ecological level, you know, it can be just a correlation that has nothing to do with what actually is happening at an individual level. But I think there are two interesting things to keep in mind here. The one is, for example, specifically for PFAS. PFOS and PFOA is decreasing, but emerging exposures to emerging PFASs like Gen X and other things, they are increasing, right? And I think that what our studies mainly capture is the effect of the PFAS mixture rather than, you know, necessarily that single PFAS that we are looking at. First, because there are some correlations within the PFAS group. And second, and there might be a lot of PFAS that we are not actually measuring, so we cannot, you know, control in our model for the effect of all other PFAS when we are looking, for example, at PFOS or PFOA. But I think what the evidence suggests is that for the mixture effects that we have across this period, there is an indication that this might have contributed to diabetes. The second thing that I think it is important to consider here is that we don't know enough about the factors interacting with PFAS. So it might be that, you know, over this period, our diets have become more obesogenic, you know, our physical activity levels are decreasing in the population, and that may be a very fertile situation where PFAS, even at lower exposure levels, can have a larger effect on diabetes through their interaction with other risk factors playing a role in this disease. Does this answer your question?
Yeah, and it actually gets to my second question, which I posted, and I'll just say it very quickly. I certainly agree with you, just because one is increasing and one is decreasing doesn't negate the effect of one on the other on that. They're just correlations. You do mention specifically, I mean, it may be people are heavier, basically. They're eating more processed foods, lower physical activities. That could be a conjecture, and I think it's quite reasonable. Those are what's for a large part increasing the diabetes prevalence in the U.S. I guess my question is to you, and again, you may not know this, but do you have thoughts back or preliminary thoughts back about how much that may be contributing to this versus PFOA or PFAS? And maybe as a part two of the question, any thoughts back with respect to where public health dollars are best spent in terms of, would it be more the obesity, the diet, et cetera, physical activity, or more in trying to lower to subpart part trillion levels in water? So any thoughts about public health dollars as well as
yeah, I mean, it's very hard to change diet and, lifestyle factors at the population level. And there have been a lot of like prevention strategies and all that. But, I think environmental interventions in this aspect can be much more effective, because it's something that you can actually intervene at the population level by acting to the source of contamination. So I really think that's a great investment in terms of dollars, and resources to tackle these environmental exposures that have a broad impact on the global population. And, in epidemiology, we know that even the effect might be smaller when you see that at the population level, it can actually, translate to thousands or millions of cases, and it can have a great benefit for public health. So that said, I think it's also very important to do more research to understand interactions among risk factors, because, for example, these results about physical activity mitigating to some extent the effect of some long-chain persistent PFAS, that's a fascinating result to me, because something that we usually say is, for example, if I'm already exposed to PFAS, I'm exposed to PFAS, and I will be exposed to PFAS for the coming at least 5-10 years. So what can I do to decrease my PFAS-associated health risks? And maybe, if I exercise more, I have some chances to mitigate some of the effect of the environmental exposure. And I think this is very important information for personalized approach that, are tailored to high-risk subgroups that can help to decrease the burden of disease in the population. But these are all excellent questions. Thank you, David.
Great, thanks. One other question in the Q&A, a follow-up on David's question. Are we able to look at any benefits from the decreases in measured PFAS, perhaps even as a mixture? If we can see an increase in diabetes incidence, we would hope to see some decrease in some studies. It looks like an intervention waiting to be analyzed. Thoughts, Dania?
Yeah, that's very interesting. And I think we have some methods, thinking about, okay, this is ISEE, right? Most of us, we're doing just epidemiology, right? But I think that we have also some analytical methods. For example, in other fields, I've seen a lot of phthalates. There are investigators that they're using, like, for example, Jesse Buckley, they're using zip computation tools, trying to estimate from observational data for other exposures like phthalates, what could be the benefit of intervening in the exposure in terms of disease. And I think that's a very interesting field, but we don't have enough information on that from the research that we have been doing in PFAS. So far, we have been trying to understand the main associations, and that's why I think now we have good evidence for main association, which just needs to go one step further and look, at the potential effect of interventions, effect modifiers, and all these other things that we don't understand very well yet.
Great. And I think with that, I think we're at time. Thank you so much, everyone, for attending. Thank you to Dania for a wonderful talk. I hope to see you all here for our next webinar, May 11th. You'll be receiving a notification about that, but please, thank you again for joining us, and thank you again to Dania. Wonderful.
Yeah. Thank you for joining us. It was great seeing you all. Thank you.
Thanks all. Thank you, Dania. That was great.
Yeah, it was very nice to
Yeah.
see you.
Yeah, nice to see you too.
So I hope everything goes well, and we see each other soon. Are you coming
Yeah.
for actually NAC?
Yes, I will be there. So yeah, we'll connect then.
So I'll see you there. That's
Yeah,
great.
sounds good.
Okay.
Thanks again. Bye.
Bye.